Okay, so beneath its murky waters, fish of the tidal Thames.
Perfect. Well, good evening, everyone. Thank you for coming along, and lovely introduction, Maria, and hearing about fish in the Thames. So as Maria said, my name is Thea. I'm a conservation biologist in the Estuaries and Wetlands team at ZSL. I'm here with my colleague, Hannah. I don't know if she wants to pop up and say hello quickly.
Hello, everybody.
So we both primarily focus on the Thames from its freshwater tributaries out to the Kent and Essex coastline. And we're usually out and about doing surveys and working with local communities. And basically the aim of all our research is to try and gather evidence about how wildlife use the Thames, so that can best inform how we continue the recovery and restoration of this very iconic urban estuary. So, next slide. Cool. So, what we're going to talk about this evening is we're going to start with a bit of a brief introduction into why estuaries like the Thames are so important. Hannah and I are then going to take you through a couple of the projects we've each worked on over the past few years and what we've learned about the life of fish in the Thames along the way. And then we're going to take a look at where we are now, and it'll be a final section on the landmark report we published last month called The State of the Thames. And then at that point, we'll wrap up the presentation and you guys can ask us any questions you might have. So the Thames, it's our second longest river in the UK. It stretches a massive 250 miles from its source in Gloucestershire out to sea between Essex and Kent. So as an estuary, the Thames drains into the sea and is tidal throughout London up until Teddington, which is west of the city. And it has a huge tidal range, it's up to 7 meters in parts. And estuaries are incredibly valuable habitats. So they act as nursery grounds for animals to rear their young in, and it's a migratory corridor for any species that need access to both marine and freshwater habitats. And this is no different for fish. The Thames is known to be home to about 100 fish species, and many of those are of commercial and ecological importance. With all of that, however, estuaries are actually one of the most poorly studied ecosystems on Earth, and that's largely because of the tidal dynamics and so the logistical challenges associated with studying them. So with that in mind, it's an ecosystem that's massively important to fish, but relatively poorly understudied. So we obviously at ZSL want to know more. And we are particularly interested in studying the fish that are most abundant, most vulnerable, and most understudied. And these are the fish in their early life history stages. So the eggs, the larvae, and the juveniles. So in 2017 and 2018, we carried out one of the most comprehensive studies of juvenile fish in an estuary in the UK. So we overcame a lot of the challenges associated with working in one of UK's busiest inland waterways. And this was a project in collaboration with Steve Colclough, who I think might be on the call this evening, and Bournemouth University. So the project had several aims. One of them was looking at developing out understanding about how fish use the estuary and how that use of the estuary and the habitat use shifts as they move from one development stage to another, so as they're growing. It was also to establish a pre-Thames tideway baseline. So for those who aren't familiar, the Thames tideway is a super sewer, if you will. So it's basically currently under construction, and it's designed to increase the capacity of the sewer network, especially when we're experiencing high levels of rainfall and the sewer network is at risk of overflowing. And then our final aim of our project was all about trying to reconnect Londoners with the Thames. And that was through citizen scientists who volunteered on our project with our fieldwork, without which the project would not have been possible. And also from lots of passers-by who would walk along the river, see us out on the foreshore and just take an interest in what we were doing, and were always surprised to hear about the variety of species we were catching, which is really cool to be able to share that with people who were just on their dog walk or having a meander, and we could let them know that, hey, this is a nursery ground for sea bass, which was always cool. So to do this, we needed a bespoke, a really simple and easily replicable approach, which involved a few different methods. So together with Steve and with Bournemouth University, we had 3 sites. We had Putney in the west, we had Blackfriars as our central site, and Greenwich further east. So Putney would, it's not freshwater, but it is further up the freshwater, the salinity gradient, and then Blackfriars as a more central site, and Greenwich would reflect more of a marine environment, so further downstream. And we did surveys at these sites every 2 weeks from March to October for 2 years. And so I said we adopted a few different methods. So we adopted 3 different methods. The mid-channel waters were sampled at the surface and at a depth of 2 meters using something called an ichthyoplankton net, which is essentially a tow, which we would made a fine mesh to catch the very smallest of the fish. And we would hold it out into the water off of the boat, into the flow of the tide, and pick up what creatures were living in the water that way. And then the foreshore intertidal zone was sampled using seine nets, and that was looking at general fish community. And the dip nets, intertidal nets, push nets, these were slightly modified to help us understand how the very extreme margins were being used by fish. So in total, we managed 512 mid-channel tows, 821 dip nettings, and 116 seine nettings, and caught 8,346 individual fish across 24 different species. Putney had the kind of greatest abundance of fish, but actually the richness of the number of species we were seeing at each of the main survey sites, Putney and Greenwich, were very similar. And I think with over 20 species found all as juveniles in the Thames, that's really solidifying our knowledge that the tidal Thames plays a really vital role as a spawning and nursery ground for fish. And our species composition reflected the saline gradient, which I mentioned earlier, of more marine species at Greenwich and then more freshwater species seen in Putney. So these 2 graphs are just showing the individual fish caught at Putney and Greenwich in each year. So the furthest left will be the Putney and the right is Greenwich, red being 2017, sorry, and the bluey green being 2018. And from this, we can see our most abundant marine species were the common goby, the flounder, and the sea bass, whilst the most abundant freshwater species were the roach, the 3-spined stickleback, and dace. Smelt was actually, which is a fish that smells like cucumber, which Hannah's going to talk a little bit about later on, is the one fish that really drastically changed its distribution between the 2 years. So Putney is a place that's previously been identified as close to likely key spawning grounds for smelt, but interestingly, we didn't find any individuals there in 2018, and that could be one of 2 reasons. It could be there was no successful spawning that year, or it could be that the spawning had moved location and timing. And the fact that we see, so the big blue, the big blue column on the Greenwich graph is smelt that we caught in 2018, and they were all at least 14 to 15 mils, so probably several weeks old at this point. And that is likely telling us that the theory behind not seeing smelt in Putney was probably that the location had shifted that year, and there are a few different environmental conditions that can trigger that, including salinity and temperature. So there's obviously a lot of data that we could talk about from a 2-year project, but I want to just mention 2 what I found really interesting observations. And one was about the flatfish flounder, which is shown in the picture here. And flounder were found in the mid-channel at Greenwich at the beginning of the year. And they were shown, as you can see in the left of the black and white image, kind of stereotypical fish position, eyes on either side of the head and swimming vertically. And by the time we were catching flounder upstream in Putney, they had morphed into their typical flatfish shape. So the eye had rotated, one of the eyes had rotated onto the other side, and their gut and mouth had also started to develop. So if you look closely, you can actually see food in the gut in the black and white image. And they moved from the water column or the pelagic zone in the mid-channel over to occupying the benthic zone or the riverbed on the margins. So that's a clear demonstration of how a fish has developed, grown, changed its habitat use to occupy a different part of the estuarine space as it moved through the Thames. And a second observation was that sea bass also did something similar, but their shift seemed to be more associated with a size change and not necessarily a specific developmental shift. So when bass grew to about 17 to 20 mil, we saw them move from the mid-channel to the margin. And in general, that's something early life history stage fish do. They're found in the mid-channel as larvae or small juveniles, and they just kind of drift in the tidal flow. And then as they develop, they start to move into the margins and utilize something called selective tidal stream transport. And basically, this is a process by which they harness the energy of the tide to move them up or downstream, and then will tuck away in kind of protected shallow waters to avoid the opposite tide. And I've just included an image here which hopefully helps demonstrate what I mean, which is from Andy Harrison's paper. Yeah, and so just demonstrating that that's likely, as they're growing up, what the fish are doing. And just to wrap up, I kind of wanted to emphasize that the 2-year study really showed to us that the whole estuary, especially the edges, is important to fish. And the Thames is a heavily modified urban estuary. Like most of its riverbanks, as I'm sure many of you know, have been developed on. It significantly reduced the amount of edge habitat available to fish compared to what we saw historically. And in some areas, there is no shallow or sheltered water on the margins at all. And the narrowing of the river makes those water flows even that much faster. And although we can't pin down the exact impacts, it's not unreasonable to assume that the loss of those edge habitats are going to be energetically expensive to juvenile fish. So for us, our priority is to looking at any loss of those edge habitats being minimised and maximising any habitat restoration opportunities available. And I'm going to hand over to Hannah, who's going to talk a little bit more about Smelt.
Thank you, Thea. Good evening, everybody. So I'm going to be speaking about a project we conducted in 2020, which was the Tidal Thames's first fish tagging study. So if I could have the first slide, please, Thea. Just to start off with a little bit about Smelt as a species. They are a relatively small species, about 15 to 18 centimeters in length as adults. And Thea stole my fun fact, they smell like cucumber. They're extremely sensitive to pollution and human impact, so have faced huge declines since the early 19th century. But the largest known breeding population in the UK is in the Tidal Thames. While they're not resident in the Tidal Thames all year round, it is an important spawning area for them. So it's thought that they congregate in the lower estuary in February and March, and then migrate upstream to find suitable spawning locations in March, April, and then back out to the ocean. Juveniles will then remain in the estuary until autumn, and then they too migrate out of the estuary. So from previous surveys that in which we've looked for smelt eggs and juveniles, we've, along with the help of other organizations and teams, have identified a spawning area in the Tidal Thames as just around Wandsworth Bridge, just upstream. So we were then interested to know if there were more spawning locations in the Tidal Thames and where these might be. Can I have the next slide, please? But why is it so important to identify these spawning grounds? Firstly, it betters our understanding of how this species uses the estuary. As a sensitive species, it's important to understand where they're going and where these important spawning areas are. And it also helps to inform and target our conservation efforts. So attempt to introduce protections to these spawning areas. This protection of such a sensitive species then has greater benefits for the Thames ecology as a whole. So before I go into the methodology, I thought it might help to just explain simply how the tagging methodology that we used works. So Thea, I'll just talk you through when to click on this little animation. So if we stop here for a second, we used a type of tagging called acoustic telemetry. And with acoustic telemetry, the first step is to deploy receivers, acoustic receivers, into the river. So that's that black box with the string attached to the buoy. And then you tag your fish. So we have a little fish on the left who's been tagged with that little black box inside. So once the fish are tagged and released, they then swim around as they normally would. So if you can click once, Thea. There he goes. And then once they get close to this receiver, once the tag and the receiver are close to one another, if you could click again, Thea, they ping off one another. And then that receiver stores that information, if you could click again, related to that interaction. So the identity of that fish, each tag has its own identification number, the time and the date, among other information. And then if you can click a couple times, and the fish will carry on its way. So that's the general idea. So next slide, please. So we deployed 25 of these receivers between the Thames barrier at Teddington, the Thames tidal upstream barrier at Teddington and Canvey Island out by the mouth of the Thames. So these were deployed between January and February 2020, and they were attached to buoys, piers, and other existing infrastructure in the river. And the receivers look like those things in the image in the top right corner. The location of the receivers is seen in the map here, the little orange dots. Next slide, please. Once the receivers were deployed, we could then tag the fish. And the fishing took place in March near Canvey Island and Cliff. And we used the help of local fishermen using a variety of fishing methods. So once fish were caught, they were assessed for size and health and whether they were running milt, which means that they're preparing to spawn. We caught and tagged 12 fish in total. We were hoping to tag more, but because of storms, we had to cancel a few fishing trips, which then couldn't be rescheduled because of COVID. So unfortunately, 12 was the total that we were able to tag. The tagging was conducted by licensed professionals from CFAS. And before this project could even take place, we had to pass through a rigorous ethics review, which we obviously got approved. And then once the fish were tagged, they were released back exactly where they were caught. The receivers were in place for 6 months, and we couldn't access the data until they were physically removed from the water. So it's not, you can't access that data remotely. You have to physically remove the receiver and then download the data that's been stored on it. So we had to wait until August to retrieve that data and do the analysis. So of the 12 fish that were tagged, 7 of these recorded detections. So if I could go on to the next slide, please. And of these 7 that recorded detections, 4 of them were particularly active. 3 of them recorded 1 or 2 and then disappeared. So these 4 were where we got the majority of our information and our data. So we gave them names for, well, identification purposes, of course, not our own entertainment. And extra points if you can guess the theme here. But these heat maps showed the main movements of these 4 fish. So I know they're not the best maps. It's really hard to see the river there, but the important thing is just to compare between the 4. So the top 3, you can see similar movement patterns, and they spent some time in the same area at the same time, which indicated to us potential, not enough data to know, but potential spawning site in that area, much further downstream than we had originally thought, because those areas on the left side of Ginny, Haggard, and Ron, that's between Deptford and Barking Creek. Harry down the bottom was much more adventurous, and he ventured all the way up to Putney. So that was the furthest upstream that any of the fish went. And typical Harry. The dates there show the first date of detection and the last date of detection for each fish. So if I could have the next slide, please. So and click a few times, please. This shows which receivers recorded detections and which ones did not record any detections. So based on this very limited amount of data that we were able to receive, we did see possible potential spawning areas, which would need more data in order to be confirmed, but it did give us an area that we can go and investigate further to see if they are actually spawning in these areas. And you see that's much further downstream than the previously identified spawning site near Wandsworth. And next slide, please. Other conclusions, we found that most fish were found to move predominantly at night. As Thea mentioned, the selected tidal stream they use the selected tidal stream transport to move with the tides. So we saw a movement that was strongly correlated with the tides. We saw they use this a lot in the Thames, more so than in other similar tagging studies. And as I mentioned before, the data suggests, though it doesn't confirm, potential spawning locations for different shoals of fish, so different groups moving into the estuary. And it also suggests possibly later spawning timeframes than we originally thought. So they could be spawning as late as later April, and fish were present in the tidal Thames until May, so much later than originally thought. So the next step to further investigate spawning sites in the tidal Thames and whether these potential spawning areas are actually spawning areas would be to repeat the tagging study with more fish this time and hopefully get a better idea of where fish are congregating and where they might be spawning. Also to investigate these areas and sample the water for eggs and juveniles, which we've done in other areas. So those are potential next steps. And besides these conclusions and these further areas to follow up with, this study also showed that this methodology, the acoustic telemetry, is really successful in a very, very busy port and a busy estuary, because that's something that can interfere with the telemetry. So we proved that this is a successful methodology and can be repeated in busy and estuaries with lots of ships coming in and out. So that was definitely a success. And that is it for me for now. So thank you very much. I'll pass back over to Thea.
Hi. Thanks, Hannah. Okay, so I'm going to talk about the European eel. And although it has quite a snake-like elongated body, it's a member of the class of ray-finned fish, and the European eel is considered a freshwater eel and belongs to the genus Anguilla. And this is a fish with an extraordinary unseen life cycle. It starts in the Sargasso Sea near Bermuda as larvae, takes it across the Atlantic Ocean to European coasts and rivers, and all the way back again. And fish that migrate from rivers or freshwater environments to the sea to spawn are known as catadromous. And European eels have a particularly complex life history. And they may be referred to as freshwater eels, but there's also a poorly understood proportion of the population that actually will never move into freshwaters and stays in estuaries and coastal waters. And so former members of our team actually at ZSL made quite a nice video, which I want to see if I can play for you. If it doesn't work with the sound, I can put subtitles on, but maybe Maria shout if this doesn't work and we can skip over.
Yeah, we're not hearing any sound. We can see the images, but we're not getting any sound.
Okay. Is it all right to do the subtitles without sound?
Yeah, that's fine if that's what yeah, we'll make sure we read. We'll read in then. Okay, so read everybody, please.
Cool. Sorry everyone for not being able to hear the sound, and some of the subtitles are a bit sketchy, but hopefully you got the gist and enjoyed the video. And it's definitely worth checking out online as well. And hopefully that showed us one of the most remarkable unseen migrations when they come right here to the Thames. But unfortunately, the number of arrivals we're getting to Europe and the Thames has drastically declined since the 1980s. So by as much as 95% in some cases. And this is decline in what is known as recruitment, and across all other stages in its life cycle actually is the reason for this classification of being globally critically endangered on the IUCN Red List. And there are several potential reasons for that decline, and the most likely thing is it's all those factors working together in combination. So those factors include pollution, habitat loss, barriers to their migration, hydropower, fishing impacts, and the variations in climate and ocean currents. And across the society at ZSL, there's lots of different bits of research going on about eel behaviour and how they move over barriers and how passes might be used to benefit them and how best to design those passes. A lot of international policy work going on, and also some local conservation delivery and community engagement, which is really what our team is working on. So what I'm going to focus on for the next few slides. So the Thames catchment comprises approximately 11% of the freshwater and lake habitat in England and Wales. So unsurprisingly, it's historically been a really important area for eels and supported large stocks. But as we already know, talked a bit about, is the Thames is also really heavily modified, and it includes lots of engineered flood defences and weirs. And it's estimated there could be over 2,000 barriers across the Thames River Basin. And these act as major barriers to eel migration and other wildlife, and likely prevent its access to suitable habitat upstream. So to learn more about elver recruitment and the impact of barriers, ZSL began a monitoring project, which was looking at elvers as they were doing the upstream migration in Thames tributaries. And we started that in 2005. And traps are placed at river barriers from April to October, which is when elver movement upstream typically happens. The warmer temperatures make eels more active. And this is quite a straightforward approach to monitoring this migration, because the eels will congregate while they're trying to find a route upstream. And this sketch here shows kind of the basic design. And you have water flowing down a trough, and it can look not that dissimilar from rain guttering, basically. And you include a media in it, which would be something like netting. It just creates a rougher surface to allow small eels to climb. And it will have a water flow going down it, and that water flow acts as an attractant. So basically, elvers will move towards the mouth of the trough, move up it, and then be held in a holding tank, which will basically keep them safe from predators, out of direct sunlight, and basically safe until someone can come along and count and measure the eels, which is done on pretty much a daily basis in the season, particularly for those really busy sites. Well, busy, lots of elvers. And the exact design varies depending on the site, because sometimes they have to be made, the nuance of each site means sometimes they have to be slightly tweaked. But these kind of broad principles remain the same. Does mean with slight tweaks in the trap design, the trapping performance, like how successful they are at eels using them, varies. So we don't we don't make comparisons between sites. We basically use it as a method to look at single sites over a period of time. So when our monitoring began in 2005, it was really limited to just 3 sites. And then in 2011, with the massive help of citizen scientists and partnership organisations, we managed to expand the monitoring. So now there's 18 sites, and 18 that have been monitored for at least 3 years, and then 5 more that have been monitored for less than 3 years, but will be continued to be monitored. And at this point, I'd like to make a sneaky mention of our Volunteer for London's Rivers page. So if any of what Hannah and I have talked about this evening sounds really interesting and you'd like to be involved and you want to be contacted about some training opportunities to become a citizen scientist, then I wanted to flag our volunteer for London Rivers form. You just have to enter your name and your email address, and you'll be added to our list. And then when these opportunities come up, you'll be told about it. And in the case of the eels, we'll have a training session where we'll show you how to safely remove them from the trap, how to measure them, and to release them upstream on their journey. So yeah, if you are interested, please visit that page. Anyway, back to the eels. So we count them, we measure their length, and we look at the trapping duration for each site, which is basically, so sometimes the traps will fail for different reasons, but we have to take an account of the days that the traps are working so that we can include that in our analysis, and we basically calculate a catch per unit effort. And that's really useful when we're making comparisons over time. And a consistent methodology for monitoring eel recruitment has been in place for a really long time for a few sites. So for river roading in northeast London, that's been going for 16 years. The River Medway towards Kent has been going for 7 years, and Molesey near Hampton Court has been going for 10. And this shows our data from Molesey over the past 10 years. And these long-term sites enabled us to really look at recruitment changes over time, but then having more sites lets us infer trends over the wider river basin. As you can see, recruitment is not particularly high, and although I haven't backdated it all the way back to the 1980s, but if you do look at some graphs like that, you see a massive crash in the numbers, and they've stayed pretty low since. We had a small peak in Europe in 2014, and that was reflected in the Thames catchment in 2013, 2014. So here for Molesey, you see the peak in 2013. But largely recruitment is mainly staying low. We're sharing this data with the Environment Agency, who share it with the Working Group on Eels, and they conduct stock assessments for the European eel. So all this data can feed into what conservation action we need to take to, and for us, the big focus is on restoring habitat connectivity. So we look at where we can install passes to enable eels to overcome the barriers to their migration and move upstream on their journey. And to date, the total amount of eel habitat that's been made accessible by the monitoring sites and the eel passes is calculated to be almost 140 hectares. So with that, I'm going to hand over to Hannah, who's going to talk a little bit about our state of the Thames.
Thank you, Thea. So for this last section, I'll just be highlighting some of the findings from our State of the Thames 2021 report and relating them to fish health in the Tidal Thames. First slide, please. The aim of this report was to show the progress that's been made in the Tidal Thames in terms of environmental health, as well as highlight some of the current challenges facing species living in the Tidal Thames. Just generally for this report, we came up with a set of 17 indicators, and these were divided into 3 sections: the state of the environment, state of nature, and state of play. And for each of these indicators, we collated existing data, and where possible, we calculated long and short-term trends in the data. So this indicators at a glance page just shows the overall summary of trends for each of these indicators. And if you're interested in reading the full report, that's available for download on our website. Next slide, please. So for this talk, first I'll just be pulling out some of the results from State of the Environment, water quality, climate change, and plastics, and how these relate to fish health. And state of nature, I'll talk about habitats, connectivity, and look at some data that we analyzed from the Environment Agency. Next slide, please. Before I jump into water quality in the Tidal Thames, it's just important to understand the London sewer system, because this is very closely linked with the Thames water quality. So I found this little graphic from the Thames 21 website, which I thought was interesting to look at and explains it quite well. So the London sewer system is what's called a combined sewer system, which means that all of the sewage from homes, businesses, etc., is mixed in with runoff from roads during rainstorms. Thea mentioned this a little bit previously. but they all go into the same sewer system. So when this reaches capacity and needs to overflow somewhere before it reaches the sewage treatment works, it overflows into the river, and that stops raw sewage from backing up into and running through the streets of London. So because this sewer system was designed in Victorian times, it's not designed for the amount of sewage that our current population creates. So this means that this overflow, this combined sewer overflow, which happens when the system is inundated, usually during rain and storms, this happens quite often. But more often than it should. So raw sewage is entering into the Thames, and the high concentrations of nutrients that are in the sewage are really detrimental to the aquatic environment. The good news, as Thea mentioned, is that the Tideway Tunnel is being constructed. It's due to be completed in 2025, and that's an updated sewer system, which is designed to handle the amount of sewage that we currently produce. So hopefully after 2025, we should see some great improvements in water quality. But with that in mind, next slide, please. The first aspect of water quality is dissolved oxygen, and that's the amount of oxygen in the water which fish and other aquatic living organisms are dependent on. And if there's not enough dissolved oxygen, which can happen especially after these combined sewer overflows, then it can cause fish kills, which has happened before in the Tidal Thames. So when we analyzed data from the Environment Agency, who collect dissolved oxygen data, and we found that there's been a real decline in the number of these drops in dissolved oxygen. So they're happening much less frequently than they were previously. So that's over the past 7 to 10 years, we've seen this great improvement. And that is closely linked to improvements and expansions in sewage treatment works. So that is a positive for fish. Next slide, please. So as I mentioned, nutrients are another part of water quality, and nutrients are high in sewage. So when sewage overflows into the Thames, it brings with it lots of nutrients, and this causes these drops in dissolved oxygen. So high excess nutrient levels are associated with poor water quality, and phosphorus is one of these nutrients. Similar to dissolved oxygen, we see that phosphorus has improved both in the long and the short term, which again is really good. And that's again due to improvements in sewage treatment works, particularly phosphate stripping, which has been introduced. And so we see a decline in phosphorus, which is associated with improved water quality. Unfortunately, nitrates have not seen the same. We've seen an overall increase in long-term nitrates, which is a deterioration in the water quality, which is a challenge for fish in the Tidal Thames. Now, next slide, please. Plastic pollution is another challenge to fish and all living organisms in the Tidal Thames, particularly microplastics. Microplastics are plastic fibers that are less than 5 millimeters, and these are commonly ingested by fish. It's not completely understood the impacts that these have on fish, but it's thought that they could have physiological damages as well as potentially could be toxic. So either way, it's not good that we're seeing so much of this in our river. A study published by Rowley et al. In 2020 found that there, at any one time flowing down the Thames at Greenwich, there are 94,000 microplastics, which is an astounding amount. And a study that was published in 2017, which sampled fish from flounder and smelt in different sampling points of the Thames, found that 70% of the flounder sampled, or over 70%, had at least one microplastic fiber within that fish. It was less for smelt, and this is due to differences in flounder and smelt feeding behavior. But still, it's really concerning the amount of microplastics in the water, but also that are being found in the fish. Next slide, please. Another challenge for these poor fish in the Thames is temperature. So temperature, we found when we analyzed the Environment Agency data for temperature in the tidal Thames, that especially in the upper Thames, so between Battersea and Kew, is increasing by quite a rapid rate. So in summer temperatures, we saw an increase of, on average, 0.19 degrees Celsius per year. And temperature changes can have impacts on fish, and this can differ by species. Some species, in some species, temperature can impact their energy, their digestion, their behavior. And we're seeing that some species are starting to adapt their distribution because of changes in temperature. So we might see more of this in the tidal Thames species, not in the tidal Thames that historically would be, or new species arriving to the tidal Thames that haven't before. Next slide, please. Just to bring up a few findings from the state of nature now. So firstly, the report focuses on the importance of conserving existing habitats, which are important nursery habitats for fish, as well as restoring historic habitat extent. So salt marsh, we found that current salt marsh extent has actually increased in recent years, which is great because that's a really important nursery habitat. Seagrass and oyster reefs have largely declined in the tidal Thames, unfortunately, but there are opportunities and hope for restoration of these habitats. Now, these three are just examples. This doesn't represent the full range of habitats that are critical for fish in the tidal Thames, but these three are quite important nursery habitats. So if we can conserve what we have and restore more, then that's great news for fish. Next slide, please. When Thea was speaking about eel, she was talking about connectivity. So connectivity is important for a lot of migratory fish species. So we looked at connectivity from the tidal Thames into its freshwater tributaries, and we marked the first impassable barriers that fish will reach on their way up these tributaries. And you can see that some of them, they can get quite far. Some of them have had fish passes installed. So the yellow shows where fish can pass without barriers, and then the red shows the first impassable barrier. Now, work doesn't need to be done, but we can see that some fish passes have been installed, and work is definitely planned for installing more. So that's a positive. It's definitely moving towards a positive for fish as people understand the importance of opening up these migratory routes. Next slide, please. So finally, we analyzed some data from the Environment Agency, who sample fish in the tidal Thames. We looked at sampling diversity, so that's the number of species per sample, and the average number of species per sample per year. So that's what's plotted here. Now, before interpreting these graphs, which looked very much like they're declining, it's very important to understand that the tidal Thames is a very dynamic and harsh environment. So if you take one thing away from this talk, it's that it's very difficult to study fish in estuaries. It's very difficult to understand fish in estuaries because it is such a dynamic and there's so many factors at play. So this section, we weren't able to come to any conclusive conclusion, for lack of a better word, because there are so many factors. So we are working with Steve Kolkoff, who I did see is on the call, and Institute of Fishery Management and Environment Agency to delve a little bit deeper into this, to investigate what might be going on here and why we might be seeing what we're seeing. Next slide, please. And lastly, just to mention quickly about some of the media attention that the report got. a large amount of the publications that covered the story focused on sharks. Sorry, I think we're a little delayed with the PowerPoint. Focused on sharks, which actually were only mentioned very briefly in the report. They weren't the focus of it at all. So we were quite surprised with that. That was something that the media decided to kind of take and run with. So we've done our best to try to correct some of the misinformation that was shared about sharks. And Thea actually wrote a blog, which these quotes are from that blog. And Thea, I didn't know if you wanted to jump in at all with any more information about sharks.
Yeah, so some people might have seen it in the press a little bit. And it's true, we do have sharks in the Thames. They are not under Tower Bridge, as some of the front covers would have you believe, but exist in the outer estuary. Have been there for a long time. We're just at the very infancy of a project trying to learn how these species use habitat in the Thames can basically safeguard their survival for the future. And there's 3 species we're specifically interested in, which is the starry smoothhound, the taupe, and the spur dog.
Thank you for that. And that is the end of us, I think. Just thank you to all of our contributors to that report. And thank you all for listening.
Thank you both very much for what was really a fascinating talk, and you covered such a lot of ground and talking about a number of different kind of surveys and different ways that you're studying the different species that are in the Thames. That so interesting that often not a great deal is known about them. And I was really struck by the saying that, you know, estuaries are a quite under-researched kind of habitat. So a lot of this is really kind of not known information, and so important if we're going to understand the best ways of conserving these environments to keep all of those species going as best we can. We are really running out of time for questions, I'm conscious. So what we'll do is we'll just take a couple of questions now. And would you mind if we gather up the rest of the questions and pass them on to you via email, and you perhaps could do a response, which then we can email out to everybody, because there's some really interesting questions that I'm sure it would be kind of nice to follow up on. So would that be possible?
Yeah, absolutely. Sorry if that's us overrunning. Sorry about that.
No, that's fine. And we were kind of our AGM, we overran a bit as well. So it's kind of, yeah, it's great. It was really fascinating. So I'm glad you covered all of that ground. And people have obviously, you know, been so interested. That's why they're asking all these questions. So what we'll do, David, is we'll just take the first couple of questions that you come to, and then don't worry about everybody else. We'll get your questions answered, perhaps just in a slightly different format. So over to you, David.
Okay, thanks, Maria. Well, there's a couple of questions at the start about the tagging project. JK asked about the relative size of the receiver to the fish, because at first glance it looked quite big compared to the fish. And more generally, Anne asked, how do you go about tagging such small fish? How do you make it work?
Yeah, those are great questions. The receivers are about this big, and they are quite heavy, but they're designed, their size is designed just to be able to pick up that transmission that is put out by that tiny tag. So the tags are only about this big, they're 10 millimeters, and that's because the fish is so small, so it needs to be small enough to fit inside the fish. And the way they're tagged is they're put into what looks like kind of they're injected through a small tube. First they're anesthetized, so they're not aware of what's going on. And then it's injected through a small tube through their mouth and into their stomach. So that's where the tag fits. And then that's where it stays. It doesn't hurt them. It doesn't impact their feeding or their life in any way that we know of, at least, and that's been studied. So it's totally approved, and scientists are happy that fish go on and live happy lives with a little tag there, and we can track where they're going.
Thank you. And let's see, there were quite a few eel questions. That's probably general eel biology. I like general biology questions. So Heather asked, what do eels feed on, and does that change as they move between the marine and freshwater environments?
Yeah, so they're quite generalist, I would say. So we'll feed on crustaceans, and as adults we'll feed on crustaceans and other fish, and also vegetation. But obviously when they are, that's obviously when they're adults, and then when they're smaller, they're feeding not like filter feeders, but will feed more on probably planktonic stuff in the ocean.
Okay, excellent. I'll do one more, Maria, if that's okay.
Yeah.
So since you mentioned this seen across Europe spike in the eel populations in 2013, 2014, Bella asked, do you know what caused that? Why that happened?
No, I wish we did, but unfortunately I don't have an answer to that. There is quite a lot of the eel science, because their life cycle is so complex, and they have both the freshwater and marine stage in their life cycles. Is really difficult to kind of disentangle exactly what's going on. So unfortunately I don't have an answer to that question.
Okay, well.
Sorry, David, you carry on.
No, that was I was just saying thank you. I'll stop there.
That's fine.